Hello students, this is Dr. Faizan Mirza. In this video, I'll be covering with you organelles in the cell, such as microtubules, microvilli, MTOCs, and I'll be discussing centrioles with you within this video, and eventually we'll be ending this at cilia and flagella. So let's start with this. Microtubule. What are microtubule? Microtubules radiate from the centrosome in an animal cell, and you can see them under an electron microscope. These are long, hollow, rigid tubes that allow as passageway for other organelles to travel. They have a lot of other functions. So one of the functions is that they allow organelles to travel within the cell from one region of the cell to another region within the same cell. The diameter of these tubes is 25 nanometer. They also provide structural support for cytoskeleton of the cell. They also give shape to the cell. What else do? They form spindle during mitosis. They are involved in beating of cilia and flagella. How are these microtubules made or they produced? These are produced and arranged. They are produced obviously being they are proteins in nature, and they are protein in nature. And what kind of protein do they have? They have a protein which is called tubulin. So this tubulin protein must have been made on ribosome or the raphia. But this protein undergoes changes in Golgi, and then it gets organized into microtubules at the centrosome or near the centrioles. So, what these microtubules are made up of? The microtubules are made up of a total of 13 protofilaments arranged around a hollow core. What do you mean by 13 protofilaments arranged around a hollow core? Let's look at this diagram. In this diagram, you can see this tube here. This one tube is one protofilament. Microtubule is made up of such 13 protofilaments. I have highlighted just one here. 13 protofilaments are arranged around a hollow core. Inside the microtubule, it's hollow, and there are 13 protofilaments all around. And the protofilaments, if you look at the microtubule from this side, so this cross section appears of this sort, 25 nanometer diameter, and each unit, which is or each single protofilament diameter is 5 nanometer. What is this protofilament made up of? Let's discuss a single protofilament. So if we take a single protofilament, so this single protofilament you can see here is made up of these rounded structures. One is blue, one is purple, then blue, then purple, then blue, then purple. What is blue and purple structure? This structure is called as a tubulin dimer. There are two type of protein tubulins which are forming your protofilaments. We call them as alpha tubulin and beta tubulin. Together they form a dimer called tubulin dimer. So how are these tubulin proteins and tubulin dimers arrange to form protofilaments? These dimers will arrange end to end. So what I have done, I have highlighted four circles here, one blue, one purple, one blue, one purple. And if I keep on going on ahead in the same way, I'm actually just adding more tubulin dimers along its length. So as a result, the protofilament will keep on getting elongated. This is what a protofilament looks like. A total of 13 protofilaments are there, and they are arranged around a hollow core. Moving on, from microtubules, we have microvilli. Microvilli is involved in increasing the surface area for efficient absorption or secretion. They are finger-like extensions of the cell surface membrane. They increase the surface area for absorption, for example, in your villi or in your kidney. Microtubules are present in the base of microvilli. They provide support to the microvilli, pretty much what they do to cilia and flagella as well. Microvilli are absent in plant cells. Coming to centrioles. Centrioles appear in light microscope as a circular structure, but in electron microscope they appear as cylinders, two cylinders at right angle to one another. So this is one cylinder, and the other is at right angle to it. You can see this circular cylinder appear to have these circular structures on its surface. What are these circular structures? Let's go to the diagram on the next page. This is what a centriole looks like. A single cylinder of centriole. The single cylinder of centriole is made up of triplet of microtubules. So there is a triplet of microtubule, which means three microtubules, and there are total of nine such microtubule sets, and these nine triplets are arranged around a hollow core. One, two, three, four, five, six, seven, eight, nine. So these nine microtubule triplets are arranged around a hollow core, and this makes up one cylinder of the centriole. The other cylinder of centriole will also be of the same sort. So there are two centrioles present in the animal cell. So this is what this structure looks like. So if you look closely, this circular structure appears to have all the microtubule triplets in the same way. And these microtubule triplets are what I was telling you. There are a total of 9 such microtubule triplets in a particular centriole, and there are two centrioles at right angle to one another. What is the function of centrioles? The centriole work as MTOC. MTOC is microtubule organizing center. This is the region from where the microtubules get organized from. Centriole, already discussed with you. Centrosome is the main organizing microtubule organizing center in the animal cell. Next, we have to discuss a structure called as cilia and flagella, and we have to just align the concept of microtubules in cilia and flagella as well. Cilia and flagella. Cilia carries out whip-like movement. They have a sweeping action. They move the structures and content which are present just on the surface of the cilia. For example, movement of mucus. Flagella, on the other hand, whip-like structure projecting from the cell surface membrane in some animal cells as well as unicellular organisms. And the movement of this flagella, the beating of flagella causes locomotion of the whole cells. The cells swim. For example, the sperms can move. For example, bacteria can swim using flagella. Cilia will not allow this kind of swimming. Cilia just is for moving the substances which are present outside the cell ahead in one direction. For example, as I just mentioned, the movement of mucus. This diagram shows to you the cell surface membrane and the extension of the cell surface membrane covering the cilium. Inside the cilium, there is a cylinder, and this cylindrical, not a cylinder, a cylindrical structure, and the cylindrical structure is having a 9 plus 2 arrangement. What is 9 plus 2 arrangement? This detail is the 9 plus 2 arrangement. 9 plus 2 arrangement means this structure present inside the cilia is actually microtubules, and these microtubules have 9 plus 2 arrangement. There are two microtubule as singlet microtubule in the center, and there are doublets which are 9 in number around these two. So a total of 9 doublets are there. 1, 2, 3, 4, 5, 6, 7, 8, and 9. A total of 9 doublets and two singlet microtubules in the center. This forms the 9 plus 2 arrangement. This 9 plus 2 arrangement is present inside the cilia from top to bottom within the cilia. The entire length of cilia and flagella is having this structure. This cylindrical structure is given a name of exoneme. The term exoneme is not not a part of your syllabus. These doublet microtubules are of two types, A and B. The B microtubule holds A, and A is having certain protein arms called dynein arms, and these arms hold the next duplet, and so on and so forth. Every doublet is held by the next doublet, and it is the microtubule A that holds the next doublet using its dynein arms. What is present just below this 9 plus 2 arrangement? Inside the cell, we have a structure called basal body. Now, when we discuss this basal body appears to have exactly the same structure of 9 triplets that we discuss in centrioles. So centrioles and basal body, they both have this 9 triplets arrangement. Centrioles are present inside the animal cell near the nucleus, and the basal bodies are present at the base of the cilia and flagella. They both have 9 triplet of microtubules present. Whereas inside the cilia and flagella, there is a 9 plus 2 arrangement of microtubule in which there are 9 doublets of microtubules, microtubule A and B, and there are two singlets of microtubules in the center. That's it from my side for this video. I hope this was helpful for you. Thank you so much.